Hello everyone, this is Athena from the Help Stop TB project, and for the next 20 minutes or so I'll give you a quick update of what we've been up to in the project lately. So first of all, a bit of information about tuberculosis itself. So the question is, what is tuberculosis? Tuberculosis is a disease that is caused by Mycobacterium tuberculosis, or MTB. It's a bacterial infection that most often affects the lungs, but it can also hit other parts of the body, such as the bones and the nervous system. It's primarily an airborne disease, which means that the way it's transmitted is through droplets in the air that are produced when an infected person coughs or sneezes. The most common symptoms include persistent cough and fever. Although there is a very common misconception that I also shared, that TB is a disease of the past, and many of us see it more like something or a scene belonging in a Victorian era movie where the protagonist holds a blood-stained handkerchief, it is unfortunately very much a disease of today. Only in 2014 it killed 1.5 million people and it infected nearly 10 million people. So a question that comes to mind now, and it's a very logical question, is why haven't we succeeded in eradicating TB yet? It's a very good question, especially if one considers how advanced medicine is today. And it's a very good question also because we do have antibiotics and we do have vaccines against TB, and they are indeed efficient. The problem is that the full eradication of the disease is hindered by three main TB challenges. The first one is that there are increasingly occurring drug-resistant MTB strains. They can resist either all or most TB antibiotics. These are the MDR, the multi-drug resistant, and the XDR, the extensively drug-resistant strains that you can see on the slide. Another problem or another TB challenge is the very long duration of diagnosis and treatment. These can take up to 3 and 6 months respectively, which increases then the possibility of mutations that can lead to TB challenge number 1, which is resistant strains. Or it can also lead, those very lengthy treatment times can lead to patients not being very willing to comply with the full course of the treatment, especially because the medicine and then the antibiotics for TB sometimes have quite severe overlapping toxicities. Another big TB challenge is HIV and TB co-infection, about which we'll talk more in detail in a few minutes. In 2015, this co-infection has led to many deaths, and in fact, 1 in 3 HIV deaths in 2015 were actually due to tuberculosis. The second question that comes to mind now at this stage is how exactly does HIV and TB co-infection works? Actually, what happens is we have a bi-directional interaction mechanism there, which means that on the one hand, a HIV patient finds it very difficult if we're talking about a case of HIV and TB co-infection, a HIV patient finds it very difficult to be able to fight TB off, as their immune system would be very weakened. On the other hand, the problem is that while the immune system would start attempting to fight TB off, what would happen is the HIV infection would get worse. So that is pretty much the definition of a bi-directional interaction mechanism. Another big problem is that HIV can have a very significant impact on how TB presents itself. As immunodeficiency progresses with HIV, what happens is that TB infection becomes more likely to start appearing in a more atypical way, which basically means that tests that should normally come back manifesting TB presence, like chest radiography for example, what would happen now is that they may appear clear, and also the TB symptoms might be mild and not very specific. Apart from the three big TB challenges that we just discussed, another big problem is the crucial challenge, the inherent crucial challenge in the fight against TB. That's the resilience of the MTB, the mycobacterial bacterium itself. The microorganism's outer coat is actually made up from some very specific lipids that are called mycolic acids, or MAs, as you will see quite often from now on the slides. Mycolic acids, are actually the first line of defense against antibiotics and against the host immune system too. What they do is that they give the bacterium impermeability and drug resistance, and what they also do is they contribute to the bacterium's pathogenicity and its ability to establish a lethal and persistent infection. What we know is that it has been shown that mutant mycobacterial strains that lack mycolic acids demonstrate an increased permeability from drugs. So if we have some mutant mycobacterial strains that don't have any mycolic acids, these ones are very much easier to be penetrated by drugs. And they were also less virulent, so they were less prone to cause disease. Mycolic acids, we know that they tend to prefer to assume a variety of folded conformations inside the bacterium cell wall rather than stay open. We also know that they have a very varied chemical nature with different chemical moieties and chain lengths. These chemical moieties, this big variety in their chemical nature has been shown to have important effects on how they fold and how they behave biologically. And this is the reason for this project, and this is the reason why we are interested in looking at mycolic acids and studying at their conformations and folding preferences in detail, because as we know they are correlated to their nature, we might be able to understand how exactly they correlate to their biological properties and their biological role within the bacterium itself. So our main goals in the Help Stop TB project is to perform, first of all, obviously perform detailed structural determination of mycolic acids, and in turn study and understand how their chemistry affects their folding. When we have done this, we would be able to assess how this folding affects their biological properties, and thus we will be able to understand more about the disease. Because we are very lucky to have partnered with World Community Grid, and we are very lucky to be helped by you, the amazing volunteers, we have such big computing power in our hands, and we are able to model hundreds of mycolic acids, and we're able to populate a big database of mycolic acid structures and get structural details for both natural occurring molecules and unnatural or synthetic ones. The data we will be collecting from our simulations, from single or individual mycolic acid simulations, will directly be useful to be fed into modeling the full membrane in the future, the full mycobacterial membrane, which would in turn give us even more realistic information about how the molecule behaves into a more natural environment. Ultimately, we could say that we are aiming to, first of all, analyze the results from the simulation, which will give us an understanding of the disease at the molecular level. From our results, we'll be able to understand more about mycolic acid's biological role, about how the microorganism's pathogenicity is correlated with mycolic acids, with their chemical nature and with their folding. We expect to be able to understand more about exactly how the defense mechanisms of the TB bacterium work, particularly how it manages to evade the attacks from drugs and the host immune system. This information will later be able to contribute to the development of new strategies that can hopefully counteract those defense mechanisms in a more efficient and targeted way. If we manage to pinpoint to a weak point of the mycolic acid structure, or to a crucial pathway of their folding that causes persistent infection, we could be looking at new strategies against tuberculosis. Now a bit of information about where I come in this project and a little bit of background information about me. So I graduated from the Department of Chemistry at the Aristotle University of Thessaloniki in Greece. After that, I did a master's in computational chemistry, which took me out of the lab and demonstrated that I loved doing chemistry on the computer. So after that, I really wanted to continue doing computational work, and I was also very curious to do some molecular dynamics simulations. At the same time, I was very interested in getting involved with a project that would study a problem that affects people and their lives. So this is when I came across the Help Stop TB project, and I was initially intrigued because, or I was immediately intrigued actually, because my first thought and my spontaneous thought was, surely TB belongs in history books rather than today's, I don't know, medicine or today's science. So my misconception made me think that a first little victory of the project would be to simply raise awareness on the disease. So I immediately applied, and here I am. Help Stop TB is my PhD project, and as I am now in my final few months, I have still lots of work to do. My role in the project so far was to lay the framework and the foundation for the thousands of parallel simulations that we are running. I had to get the simulations up and running, and this had to be done in an efficient way, which required lots of technical work. First of all, I had to decide which mycolic acids to build and model, so I had to consult the available literature in order to decide what the modeled systems would be. After this was done, the systems had to be set up and systematically established so that they would be able to run both on our platforms and on the World Community Grid systems. After this completed, I needed to do some initial configurations and calculations. This is basically the most important part. The preliminary molecular dynamics is the most important part of this work, because this is where the systems are getting equilibrated in both temperature and pressure, so that they would be able later on to run smoothly during the data production runs and avoid blowing up, as we say, and crashing. For this, I established some permitted energy thresholds that the molecules would be allowed to explore. And I also established an automated workflow for this process, because obviously this couldn't be done by hand because they were thousands of them. This automated workflow process resulted in thousands of equilibrated simulations completing on the University of Nottingham's high performance computing facility and being ready to pass on to the World Community Grid for data production. Then we set up our server with World Community Grid, we were set up and running, and there was when I had to establish an automated data handling workflow so that our simulations would be able to run efficiently and, most importantly, automatically. On this little schematic that I have there on the slide, you can see how we build up from our initial 166 structures. We performed some initial configurations from where we extracted 100 frames from each molecule and then performed solvations, minimizations, and 2 different equilibration runs before proceeding to the production runs. So this is where we are now. Our current status finds us nearly 4 months after the launch now, with more than 900,000 results returned. Thank you all so much for the amazing contribution, because it wouldn't have been possible without you. Our next step, crucially, is to develop automated workflows for the analysis. This will allow us to look at those thousands of simulations and identify differences in the folding between different mycolic acids. And in turn, this will directly help understand more about mycolic acid's biological role within the Mycobacterium, and ultimately lead to more understanding and more knowledge about the disease at the molecular level. The tools we are currently using to look at our results are based on statistical models that will help us reduce the dimensions of our data, as we say, because we will be receiving back so much data. We need a way to reduce the dimensions or reduce the sheer volume of it in order to be able to look at the most significant and most important information in our data. The method we're using for this is called principal component analysis. It's a statistical procedure that compresses a whole mountain of data into something that captures the essence of the original data. As the name suggests, principal components, we strip off the less important information and we focus only on the very important one. The analogy that I read online when I was trying to understand what principal component analysis, or PCA, was about, that I found very useful to understand, was exactly how a camera records actors, buildings, objects, and then the movie at the cinema is kind of flattened and projected in 2D without us losing too much information. This is exactly how PCA works. In order to achieve this, we are using and adapting PCA tools that were developed at the University of Nottingham and the University of Portsmouth. So our outlook for future work surely includes the further development of an automated workflow that will be able to handle and analyze all the resulting data in a meaningful way. This is definitely our first priority at this stage. A future task that is directly derived from the analysis of our results is to feed these results, to feed our conclusions into building the model of a full membrane, and ultimately in the future model the complete mycobacterial cell wall. Another task we will be getting our hands on in the near future is to model interactions of mycolic acids. So instead of performing molecular dynamics simulations of single mycolic acids, we will be packing more than one closely together. We will be trying to pack 5 or 10 different mycolic acids closely together in order to simulate how their interactions affects their folding, and we can see what exactly this interaction translates into. We would also be interested in performing molecular dynamic simulations into which we would be looking at drug and maybe mycolic acid interactions. So to finish up, I'd like to thank you all first of all for your attention through this little presentation. Hopefully you enjoyed the little update on the Help Stop TB project. Much more importantly, a big thank you to all volunteers who have kindly donated your computer time to our project, and to all World Community Grid projects. I know there are many exciting projects available. So for one more time, thanks again from the whole team here in the UK, and goodbye from me.